Welcome everyone to attend today's our last Monash Biomedical Imaging webinar with Professor Gary Egan. It's our last seminar, and we're lucky to have Gary to give a summary of Monash Biomedical Imaging activities, but I also think he will give his vision on how to extend Monash imaging landscape in the new year. So really looking forward to his presentation. So just a housekeeping item. Please ask your questions for the speaker via the Q&A button, and these answers will be answered at the end of the speaker's presentation. The chat function is disabled. So before we start the presentation, I would like to acknowledge the people from the Kulin Nations on whose land Monash University is positioned. I pay my respects to their elders, past and the present. It's my great pleasure to introduce our speaker today, Professor Gary Egan. So Gary is the director of Monash Biomedical Imaging. He's a professor at Turner Institute for Brain and Mental Health. I guess I don't need to introduce too much on Gary. He has made quite impressive contribution to the Australian neuroimaging landscape. And in particular today, he will talk about the activities of the MBI. So I hand over to you, Gary, and really looking forward, and thank you for your time today.
Great, thanks very much, Iain. I'll start sharing my screen, and hopefully that will enable you to see my slides. Are you seeing my slides full screen, Iain?
Yes, that's good.
Okay, great. Well, thanks a lot, Iain. It's really a pleasure to give a wrap-up seminar for MBI this year. It's been a big year, as most years are, and we've got lots more to come in the future. I always like to start with this slide. It gives a very sort of compact overview of the facilities and expertise and capabilities we have at MBI and at the Alfred Research Alliance MBI node. I think many of you will have seen this previously, but we have had some good progress this year, which I'll speak about during my presentation. In particular, further development on the MRI-guided focused ultrasound development, which was initiated a couple of years ago. We've had a very successful establishment of the magnetic particle imaging system at the Arambi node this year, and that's underway. And we've made great progress on the establishment of the cyclotron facility, which will come online in a couple of years' time. And I'll give more details around that at the end. So I'm going to give a summary and an acknowledgement to the teams and the link labs based here at MBI. And I want to start with the management and administration team, led by Kylie Reid, and you can see the other team members here. I really am incredibly indebted to them for their great work. You know, through the COVID lockdown, it's been a real challenge to maintain research imaging services, and the management and admin team has done that, you know, absolutely admirably. In addition to that, they've also collectively worked on establishing a FEED study for the cyclotron project, FEED meaning the feasibility and engineering design study, which is shown here in the top right, conducted by a company called FarmOut for us earlier this year. That was a very significant piece of work. And then more recently, working with two industry partners to put together a proposal for a further expansion of the cyclotron facility that I'll talk more about later. The administrative team has also done a huge amount of work this year to establish an S9 poison license for procedures and training and then application here at MBI in relation to the psychedelic research work that's getting underway. I'll speak more about that. And of course, the team supported the large-scale human imaging projects to get back on track after reopening a month ago. And we really look forward to intensive activity over the next 12 months for researchers to be able to catch up on lost time. And you can see here a list of the studies which are re-established and ramping up and really going to take take the full availability of the human scanners. The COVID policies and procedures were put in place continue to be put in place this year by the management and admin team. And in addition, there's been some further streamlining of procedures, including OHS induction activities, which are really very, very valuable. And I acknowledge James and Louise in that work, and of course Janelle before she went on maternity leave. It's been identified across many university OHS departments as really an exemplar approach. So we really are very fortunate that we've now got a very streamlined induction system, which I did myself again a week or so ago, very efficient. And we've also been continuing to develop what's called the Imaging Research Identity System, or IRIS. And this is work that Parissa in particular has continued on with together with Shenzhen. And this is a system now which, as well as providing identity management, also provides access to images for radiologists and nuclear physicians to report images remotely and to record those reports and then to flag cases where there might need to be follow-up. So this is something that's being looked at for other national imaging facility sites. And it's great that we've initiated that here at Monash, and it's now being looked at carefully for application elsewhere. We had a PQMS external review, which was undertaken and passed with flying colours. And as we'll hear shortly, we launched the world's first technology with MPI hypothermia treatments and CT imaging at the Arambi node. And here it is, a great achievement this year that a world first imaging instrument was, the installation was completed back in, I think it was probably April, May. It was a challenge. We had delays, of course, due to COVID, difficulties of getting engineers in. And this instrument is the world's first configured magnetic particle imaging system with a CT scanner and an associated hypothermia unit, which can then provide treatment regimes targeted at magnetically labelled particles in vivo. And you see here in this photo, Karen Alt, a senior research fellow at the Central Clinical School, setting up and using the magnetic particle imaging system. It's for mice only. And you can see here on the right, the first image that came off from a live mouse back in June, and then was put on the cover of the newsletter for Imagine Biosystems, one of the key industry partners with the MPI researchers at Arambi. So that's been terrific. We've actually had an experiment done by Karen and her colleagues. Not quite finished. We had a little hiccup with the CT scanner on the unit. Being the first in the world, it's not surprising that there'd be some technical hiccups, but hopefully that will be resolved very soon and we'll be able to continue that experiment and get the first completed and published study out early in the new year. Other achievements at Arambi have been very significant around utilisation of a PET CT scanner, a massive increase in the utilisation of the facility, as well as grants that have been received by Bianca Jupp, Lucy Vivash, and also Terry O'Brien, of course, with the development of new agents for looking at neuroinflammation. So this is really exciting. We're delighted that we're able to support their new development, radiopharmaceutical development work in conjunction with industry. There's also a greater provision of radiopharmaceuticals, both at Arambi and here at Clayton now, by coordination between the two sites. And Rob Vajelka has been instrumental in setting that up, including access to isotopes at the Alfred Hospital Nuclear Medicine Department. So that's really expanded the utilisation and the opportunities for using the instruments. There's going to be a greater emphasis going forward on the use of copper-64 label compounds, and the first work with that in a copper-64 label ATSM compound, traditionally used for looking at hypoxia, is now being used by Iain Harding and colleagues and others to look at oxidative stress in neurodegenerative diseases. So that's a really important development, both for preclinical studies as well as human studies. So a little bit about the preclinical team at MBI Clayton. led by Mike De Beer with Gang and Katie and Hong, who supports them. And you can see here their respective expertise on the PET spec CT, the 9.4 TMR, and the ultrasound and focused ultrasound unit at Clayton. So the group's been very active this year and achieved a number of important milestones. You can see here on the left that there's been great progress in establishing a cardiac MRI imaging protocol for looking at fibrosis in the heart, and first results of that have been made available to the users. Christian Samuel and colleagues, and other studies are underway. There's been continuing a significant use of the focused ultrasound unit in collaboration with colleagues from CSIRO, and particularly Lars Ittner and Nico Volker. And that's been involved in establishing and validating an in-house glioblastoma tumour model, which is now underway with the first experiments being done later this month and in the new year. So that's been really significant. We're really looking forward to the results of that focused ultrasound work. There's also been a couple of major equipment grants that the preclinical team put together for a new PET-CT, a new high-resolution CT, and a new photoacoustic ultrasound system. So we're waiting on the outcome of those grants from the ARC large equipment at the moment, and look forward hopefully to getting that result before the end of the year. Looking ahead, I think the preclinical team is going to continue to focus on these key areas, looking at cardiac imaging protocols with particularly MR and ultrasound, looking at the use of the focused ultrasound to manipulate the blood-brain barrier in animal models, and then look at therapeutic interventions. And in addition, there's a significant work program around improving kidney imaging in preclinical models using susceptibility-weighted approaches, and also the development of new PET-labelled tracers for looking at in vivo large animal and ultimately human kidney imaging applications. So that's a very important piece of work in being led by John Bertram from BDI. So here's a couple of examples. This slide here shows the results of the T1 mapping work that Gang has done in conjunction with Mike and Christian Samuel and colleagues. And David Wright's been very critical to the development of this work, which enables quantification of very diffuse areas of fibrosis in the myocardium. And importantly, it's been validated using histological approaches. So this is now very ready to go in cardiac studies in mice. Another study which the preclinical team undertook with an industry partner this year was with a company called Adelta. It was a very important study to look at the biodistribution of a zirconium-89 labelled tracer to look at fibrosis and inflammation in idiopathic lung disease. And I really want to acknowledge Mike De Beer and his team for undertaking this work, and of course Kylie for managing this project. It was an important project that identified that the route of administration of a new drug with IV would not be practicable, and the company has used this work now to redirect their energies to looking at a inhaled delivery system for the drug to increase the availability in lung fibrosis patients. So this is a great example of our key, not just our capability, but also our ability to, in a timely fashion, respond to industry's needs. The whole project was undertaken in about 6 months, which is pretty impressive in the context of looking at a macaque non-human primate study. So that was a great step forward for the preclinical team at Clayton. David Wright's lab at the Arambi node has also been really active. The scanner there is incredibly busy, and David's lab is growing with research grants and fellows. And I want to put this slide up, which he shared with me, around the remarkable results that they're getting looking at traumatic, mild traumatic brain injury and concussion in footballers, and the use of the very sophisticated diffusion MRI white matter imaging techniques, which have been developed in collaboration with colleagues at the Florey. So he was published this year in cerebral cortex, and of course, you know, it's very, very important that this work is brought into the context of assessments on the field and decisions made in relation to players continuing or not to play in high contact sports such as AFL. David's group has also recently got a FightMND grant, which is going to expand the use of the scanner there of course, in motor neuron disease. And in particular, there was a National Imaging Facility grant to David and his team to establish an in-bore focused ultrasound system on the 9.4 Tesla scanner at Arambi, which will be very significant in being able to then manipulate therapeutic interventions in neurological diseases. So that's an exciting extension and will be complementary to the focus ultrasound facility here at Clayton. David also chaired or was convener of the Victorian Biomedical Imaging Network meeting held a few weeks ago. It was supposed to be face-to-face at the Alfred. Unfortunately, we couldn't, but we had over 130 attendees by Zoom, which was excellent. And it was a really great meeting, which portrayed the strength of biomedical imaging across Victoria and across Melbourne. So David was also the recipient of the Emerging Leader Award last year and gave a presentation at that meeting, along with Katarina Voigt from here at Clayton and a number of other colleagues. So we're looking forward to running that event next year, again at Arambi, but this time as a face-to-face meeting. So I want to move on to some of the other teams and start with the image analysis team led by Xiaolin. Xiaolin's team, you can see here, is growing, and there's a number of new PhD students who have joined, including Cameron and Mevan and Hamashi, who's co-supervised with Monash Heart in the Faculty of IT. We're really delighted to acknowledge that Anjan has completed his PhD this year and submitted his thesis for examination and is heading off overseas in the new year to take up a postdoc position. So we're really delighted he's got an appointment at UC Davis. The group's output has been really, really impressive for 2021. Over a dozen peer-reviewed and conference papers, a couple of patents going into the national phase, a major grant for the establishment of a low-field mobile MR network, which I'll speak to briefly, the continuation of a discovery project grant that was awarded to Xiaolin last year, and an ongoing linkage grant. There's been a lot of recognition of the research done by Xiaolin's team, particularly Viswanath Sudarshan, who was a PhD student, finished late last year and was conferred earlier this year, was the winner of the Award for Excellence for PhD at the Institute, Information Institute and Technology in Bombay, where he was jointly enrolled between IITB and Monash. So that was a great recognition of the excellence of his PhD work. We also had Kamlesh, his work looking at brain image segmentation using brain images of brain tumours was placed second internationally in a challenge conducted by MICI, the image analysis community's annual international meeting. So that was an outstanding achievement and really a credit to Xiaolin and Kamlesh in the excellence of their work. So a couple of examples from the image analysis team. You see here work on the top left from Viswanath's latest paper in medical image analysis, looking at the ability to do low-dose PET imaging using MR-guided anatomical priors, as well as then a deep learning model on the reconstruction of the PET images. That's really exciting work. The top right, you see their work that's been done and published this year from the group around joint estimation of fMRI bold signals and PET metabolic activation signals. That's been very well received, and it's really set the scene for a whole range of research programs now, which particularly Shana's group is undertaking. And on the bottom right there, you see the work that Anjan has published from his PhD, looking at the localization of iron in the precentral gyrus in motor neuron disease patients, and then quantifying this using susceptibility weighting and quantitative susceptibility mapping techniques. So a really lovely piece of work, and great credit to Anjan and his PhD work. I mentioned that the image analysis team has also had a grant for a low field mobile MR network, and this is what it looks like. It's a very low field, 60 millitesla, so normally we're working with 3 Tesla MR scanners. This is a very low field, safe. You can have it on a regular room, it doesn't need shielding, and you can see in this case it's currently being used for primarily brain imaging. It has future application for looking at limb and extremity imaging. And it's a novel approach that Xiaolin has proposed, to use a federation of 4 scanners of this type around Australia to combine data and improve the quality of the images using AI methods. And you see here in the top right an image with 5 different contrasts acquired with the hyperfine scanner, compared to at the bottom a 3T image data set. And you can see the images are clearly way inferior, but remarkably good when you think that this is a mobile unit, albeit 500 or 600 kilograms, but still able to be manipulated and moved around within a ward and made available in rural and remote locations. So it's very interesting concept to make, essentially democratize the availability of MRI out into the community at point of care in either primary healthcare environments or in major hospitals and wards and departments where you wouldn't otherwise readily be able to access, have patients access scanning. So it's a great, exciting project, and we look forward to that getting underway in 2022. I want to turn now to Shana's team, the Cognitive Neuroimaging Lab, and you can see here it's growing very substantially. Lots of new team members and lots of great research outputs. It's got 2 main themes, looking at functional and metabolic mapping and connectivity measures in aging research, and secondly, the cognitive neuroscience of science of parenthood, which is fascinating. The group's been very active this year, as you can see from these papers, and in particular the data papers that Shana's published have been very highly cited. The data sets have been downloaded many hundreds of times, and just recently the first 2 papers using those data sets, open data sets, have been published by other groups. So it's really very pleasing to see the interest in the research data, and then the uptake of the data for subsequent analysis by other groups. There's been a strong output as well in the parenthood projects, with Winnie Orchard completing her PhD, and congratulations to her. She's also heading off in the new year over to Yale, so that's really terrific. And a lot of other work that's now coming out with particularly Katrina, who's picked up on the simultaneous MR PET work in aging to look at new ways of correlating cognitive measures and integrated cognitive scores together with the metabolic and functional mapping. I know there's a big plan for 2022 to collect more data. I think the data pipeline is almost empty. It's got to be recharged. And so that's what's actually happening at the moment. The studies are getting back underway, and we look forward to seeing some of this work being made available in 2022. Iain Harding's group is jointly between the Central Clinical School and here at MBI. And Ian's focus is on looking at mechanisms of neurodegeneration. His group has also grown very dramatically, as you can see. And his output this year and his team's publications have been really, really fantastic. One long-standing paper, or a paper in Annals of Neurology this year, from a long-standing collaboration that was set up with the Enigma Consortium some years ago, which Iain has led as the Enigma Ataxia collaboration. And really pleasing to see this beautiful work published this year. There's also been work that Iain's pioneered, actually, looking at applying a PET imaging agent called FEMPA to look at neuroinflammation in Huntington's patients. And he has also studied in Friedreich's ataxia patients, and that was published this year in Movement Disorders. So you can see a very important approach to look at mechanisms of potentially neuroinflammation in early and mid-stage Friedreich's ataxia and also other neurodegenerative diseases such as Huntington's. Iain's group is got a pretty active program looking forward, as well as those traces. There's also the copper 64 ATSM work, which will be being undertaken preclinically at Arambi and with the human studies here in Clayton. This is really a tour de force, and it sort of motivates why we're getting a cyclotron facility in radiopharmaceutical manufacturing here in Clayton. The challenge is to get these traces from other cyclotron sites nationally, because they're not available as a routine commercial product from either Cyclotech or GMS or other manufacturers. So at the moment, Iain's trying to source this from SAMRI in Adelaide, and that's been really challenging, and it's greatly limiting the ability to undertake molecular imaging research studies with traces in cohorts that are where it's a new indication. So that's work that's going to be very active, I think, in 22. And also, of course, the continuation of the Enigma Consortium work that Iain's been leading. I want to turn across now to Professor Abdulrazi's lab, the Systems Neuroscience Laboratory, Computational Systems Neuroscience Lab, which has also grown. All the labs have grown. I want to acknowledge Adil's appointment as a Saifa Azurali Global Scholar this year. That was a really outstanding achievement. And he's been appointed as one of just a handful of global scholars by the Azurali Foundation based in Canada. So that's an enormous achievement for Adil. And of course, it's because his research is absolutely flowing. The brain modeling output and phenomenally innovative and sophisticated approaches to understanding how one can model complex signals from multiple imaging approaches, obviously MRI, increasingly EEG. He's also looking with us at some of the metabolic mapping data sets. So that's really very, really having an impact, as you can see in the literature. And in particular is working on a project with Devon Stoliker, one of the PhD students with Adil, looking at altered states of consciousness, and in particular with the psychedelic psilocybin. So this comes back to some of the earlier comments I made around the establishment of the ability to image, to have S9 poisons on site, and then to undertake research with psychedelic compounds. So Adil's and Devon have set up this first project in normal subjects to look at essentially altered changes in the brain connectivity during the effect of the drug, as well as under meditation status. So this is a really fascinating study. It's a large study, and Devon and Adil are currently recruiting. So I think that's going to be interesting to see who would like to be involved in the study with psilocybin and the psychedelic experience. This work's really only happening because BrainPark is based together with MBI, and Beck Seagrave has been running this whilst Murat's away for a while. And I know Beck and her team have done an enormous amount to take the BrainPark facilities essentially online, both last year and this year, which has actually led to increased utilisation this year compared to last year. So that's really fantastic to see. The work that they've done together with the Eternal Adult Psychology Clinic to look at brain gains as a neuroscience-based approach for brain and mental health has had 2 successful pilot rounds and is now being looked at very carefully by the St Kilda Football Club with support from the Denny Frawley Wellness Centre. So we hope that that comes to fruition. We've also seen a part of Brain Park turn into essentially a medication room, which is related to the psychedelic research. And this is a really exciting development, both for a deal study that I mentioned, and also in particular for a new program of work, which Murat and Paul Lignatsky and Suresh Sundram are undertaking here at MBI and at Brain Park. And this work is essentially a world first to now start to look at the potential therapeutic benefits and the ability to use psychedelics as treatments in mental health. It's a very important area of work which has grown rapidly through Murat's invitation to Paul to come and join here at Brain Park and at MBI, and we're really delighted that that's happened. Paul's got a very large study funded by industry, which is going to start in the new year, and it's to look at the potential therapeutic benefits of psilocybin in generalized anxiety disorder. So this will use the uptake and medication room at Brain Park and potentially further spaces that we'll be developing in the ground floor area of NBI to accommodate this type of work and future clinical trials that Paul and his team have got planned. So that's a very exciting development, and we're really delighted that it's a joint NBI and Brain Park initiative. Alex's team, the Neural Systems and Behaviour Lab, continues to power on, and it's great that people have got back into the NBI facility after being away for such a long time. Great to see everybody back around again. Alex's group has been, as ever, super productive. Over 30 papers during 2020, 2021, and you see a number of them highlighted here. There's also been many awards to the people, group lab members with Alex, and in particular Sid has got a number of awards through both the BPA, the Biological Psychiatry Australia award for the best abstract, and also an OHBM Open Science Fellowship. So we give our congratulations to Sid and all of the members of Alex's team for their achievements in 2021. Looking ahead, I'm really pleased that Alex has got the Monash Brain and Behaviour Project at the top of his list to get going again in 2022. Looking to scan over 300 people, I believe, which would be fantastic. And of course, in particular, accessing large data sets elsewhere, such as the UK Biobank and the Human Connectome Project, both development and ageing, to continue doing these large-scale population-based studies of brain structure and connectivity and network properties. Adrian Carter has been at MBI a few years, but now he's got his own independent linked lab, which is a Neuroscience and Society group, which is really exciting. It's a great program of work, and we're delighted that Adrian's work has bridged across with Murat as well as from the ARC Centre of Excellence, and has got a broader base of activities, as you can see here, and a very large and growing team. In particular, the link to the psychedelic-assisted therapy, I think, is a really important aspect of how some of these parts of MBI and Brain Park are now able to come together and really provide a capability to do these clinical trial studies, which just doesn't exist anywhere else in Australia and probably in just a few places around the world. So we really have created something here which is now recognised as being unique and is giving researchers great opportunities for research, but also partnering with industry who want to look at the potential commercialisation opportunities with these long-standing drugs. So Adrian's team has achieved a huge amount this year, as ever, with over 20 papers and many great conference presentations. In particular, I just want to acknowledge that Adrian himself was appointed as the editor-in-chief for neuroethics this year, which I think is testimony to his standing in the field internationally, and also the recognition of the importance of this field going forward. We saw this in the early days of the Centre of Excellence for Integrative Brain Function, that the actual reach out to community and the emphasis placed on understanding communities' attitudes and perspectives towards neuroscience research, and particularly devices, neurodevices and neuromodulators, was really critical. So that's really important for us to keep supporting. There's a lot of plans from Adrian's group for 2022, and in particular, again, just highlighting there the medicinal cannabis and the look at clinical psychedelic trial work here is going to be really great to see the fruition, the benefits from that. I've got a couple of sections to finish. The first is I want to acknowledge the ARC Centre of Excellence for Integrative Brain Function team. We've had a centre based here at MBI for the last 8 years, and it's now essentially finished, which is a little bit sad, but all good things must come to an end. The centre has actually had a really great run of research and collegial activities, and that's all reported, as you can see here, in our annual reports over the last 7 years. We've been continuing through 2021 with a 1-year extension by the ARC, and I do want to acknowledge the central team members who have kept the centre moving along, and that's been with Glenn Papworth until he left back in the middle of the year, and Marsha Perry took over as the centre manager. So we're delighted that Marsha and Jess, Merrin and Hutch have been able to sustain the centre through to the end of this year. You can see here in the top left, the centre and the executive has continued to meet throughout the last 2 years by Zoom, which, you know, as everyone has been. But last night we were able to have our state-based end of centre celebration event down at the Yarra River. So that was a really great thing, that we did have a small window of time to get together and have a celebration around the successes of the centre as it's now finishing. And here you see some of the outputs from the centre as reported in last year's 2020 report, and we'll be having the final report done through to March next year. So finally, where are we looking forward to in the new year? There is a major redevelopment happening on the MBI site in 2 parts, actually. One is around the Australian Precision Radiopharmaceutical Facility, and the second is around the Victorian Health Innovation Centre, which you see here in the bottom right of this screen. So that was announced a couple of months back by the Vice-Chancellor, a Victorian State Government grant to build in the location where the current lecture theatre is, a new 4-storey building to essentially help drive the innovation and industry partnerships with the Victorian Heart Hospital across the road. So that's a very exciting development. We really look forward to that getting underway. I think probably sometime in the second quarter, there'll be works commencing. The architects and engineering consultants have all been appointed, and they're now commencing user engagement meetings. So the project's underway. I think the Health Innovation Centre, or maybe renamed as the Heart Labs, will be actually close to completion by the end of, well, perhaps early 2023, whereas the Precision Radiopharmaceutical Facility has got a longer gestation. And you see here some of the detail of the design. So that's the area in the back of the site, adjacent to where the current reception area is, where there'll be a cyclotron facility for R&D purposes established, and enable us to then produce radioisotopes and radiopharmaceuticals in-house using a medium to high energy cyclotron. So we've got detail done through the FEED study towards this goal, and it's going to be not just addressing our R&D needs here at Monash, but also more broadly nationally, because we've included a capacity to produce GMP manufacturing of pharmaceuticals for late-stage clinical trial work. Whereas at the moment, you can only really undertake non-GMP produced traces with for phase 1 studies. So that's a really exciting development. It's a big development. It's going to enable us in the first phase to produce fluorine-18, carbon-11 and copper-64 labeled traces, consistent with our current projects. And then in a subsequent phase, we're looking at zirconium-89, potentially iodine-124, and some other compounds. So this is really an important development effort for our research platform, and we hope that we'll have a facility up and going by sometime late 2023. Unless actually we get another addition to this facility, which we've recently made an application for, which is the commercial manufacturing of radiopharmaceuticals using this plus an expanded cyclotron, second cyclotron capability and expanded manufacturing laboratories. So this is a grant currently under consideration by the federal government through the Modern Manufacturing Initiative scheme, and it's led by Global Medical Solutions together with Telix Pharmaceuticals and Monash University, where the facility would be located. So in a nutshell, just showing you the layout of the ground floor of the facility, all of the areas on the right here would be new expanded areas for manufacturing under GMP conditions, traces for commercial use elsewhere in the country and even for exporting to the Asia Pacific region. So we're very, very excited about the possibility of this grant, and we'll keep our fingers crossed until just before Christmas when we expect to hear the outcome. It's a network, it's an application which has got a real strong network of supporters. In addition to the industry lead and partners in Monash, we've had very strong support from healthcare networks, from our international research partner in Helmholtz in Dresden, as well as the nuclear medicine community nationally, to really address a national need around the need for a 30 MeV capability cyclotron for producing long-lived radioisotopes for SPECT and other applications. So we wait to hear the outcome of that grant. So that's the summary from myself for this year, and looking forward for 2022. I just want to say again, thanks to all of the MBI staff for their terrific efforts throughout 2021 through the COVID lockdown. It's been, a tough year. We've all come through it, and we're now looking, I think, poised well to look to 2022 to get back to full activity levels and to step up into the new opportunities that are presented through many of these different recent grants and grant applications in the mix. I also want to acknowledge our new Pro Vice Chancellor of Research Infrastructure, Jack Janasek, who's just come on board as head of the Research Infrastructure portfolio. We really look forward to working with Jack and the new research leadership team that Rebecca Brown has had appointed. So thank you very much for your attention, and delighted to take questions from people.
Great, thank you very much, Gary. I demonstrate the breadth and the depth of biomedical imaging research at Monash. It's really great to see at the end of the 2021, we have new projects coming up after the whole pandemic lockdown. So are there any questions from the audience? I do have two questions, Gary, I want to ask you. In the end, you mentioned the two major investments, right? The Victorian Health Innovation Center, as well as the Australian Precision Radiopharmaceutical Facility. For the former, how the imaging can play in the Victorian Health Innovation Center? Do you think how biomedical imaging will fit into the whole health center?
Yeah, look, the Health Innovation Centre is primarily with office space and facilities for external partners, industry partners, and that to be located here and to be co-located with the Heart Hospital. But we think many of those companies would be interested in undertaking trials, looking at cardiac devices, for example, and that imaging, whether it's in preclinical models, large animal preclinical models, or then maybe human clinical trials, would be really important there and a great capability to be able to offer those sort of companies. And of course, there's the preclinical imaging capability that I described, which researchers, Kirsten Bubb and others from the BDI are now using MBI's rodent imaging capabilities in cardiac studies. So I think that's a really great capability to offer to companies. We do, though, probably have to expand our large animal imaging capabilities. in terms of holding facilities and longer term holding than what we currently have. The large animal work is mostly with animals brought in on the day and then taken out afterwards. So that's one of the new potential areas of further development we might need to make.
Yeah, great. Thanks, Gary. For the Australian Precision radiopharmaceutical facility, what would be the first tracer? What would be the first project you envisaged?
Yeah, so that's a good question, and I'm really pleased that you've asked that. We will have a number of possibilities. It'll be, I'd say, almost certainly an F18 label compound, and it could well be something very simple like sodium fluoride, which is a good myocardial perfusion imaging agent, probably FDG, and then we'll probably look at, a compound that most users can use in a kind of in a coordinated fashion, but a dopamine imaging agent has been discussed for some time. So if dopa is a possibility, or maybe something more specific such as F18 labeled falipride, looking at D2 dopamine uptake would be a possibility. And of course, the copper-64 work I mentioned would be another area. So we can't do everything at once, but we'll have a lot of discussions with research users to try and prioritise which traces and then which actual projects can use these different traces in a coordinated fashion.
Yeah, great. We're really looking forward to see the facilities going forward in the new year. Well, in view of time, I think that concludes our 2021 seminar series, and thanks everyone for coming in, and thanks Gary for today's excellent summary, and looking forward to another year at MBI, and thanks everyone again. Oh, we do have a question from David Walker. So before we conclude, David is asking, what capacity do you have for the functional near-infrared spectroscopy? My group at the RMIT University recently acquired equipment for adult and newborn work, and we would welcome the chance to collaborate with MBI.
Yeah, well, thanks very much, David. I'm delighted that you've been on for the seminar. We don't have a near-infrared imaging or spectroscopy capability, so we'd be really pleased to look at how you want to use that and whether or not there's any synergy with the way we're using MRI and EEG. I'm presuming this is a human system for adults, but it could be a neonate system, and we'd be really interested to talk with you about just how that could be integrated and made available to researchers that can increase the access and use of such an instrument.
Great, thanks Gary. Also thanks David for the question. Okay, everyone, thank you very much for your coming for today's session, and thanks Gary again for your seminar. See everyone for next year. Thanks.
Great, thanks, Alan.